Hi, my name's Christopher J. Ward from the Kansas University Medical Center, KUMC. And today I'm going to talk about DNA quantification. Now, measuring the amount of DNA you've got in a sample is one of these basic techniques that you must have off pat if you're going to be a molecular biologist. Now, in the old days, to quantify DNA, we basically just ran agarose gel, and we would slot on a molecular weight marker, say about 1 microgram, and run that out, and each of the sub-bands in that DNA molecular weight marker would be about 100 nanograms. And we'd also put on our various samples and run them out. And that basically told you, one, have you got any DNA? Is it intact? And roughly how much you had. Then along came the nanodrop, and we used the nanodrop for a long time, where you take 1 or 2 microliters of your sample, and that gives you a optical density 260 and a 280 ratio, which gives you an idea of how pure your DNA is. The 260 tells you how much there's there. But in our hands, this wasn't really reproducible. The results were kind of variable. And we needed a reproducible assay so that we could do our transfections, transient transfections and stable transfections, where we're taking plasmid DNA and putting these into cells. So Invitrogen came up with a new assay called the Qubit double-stranded DNA broad-range assay kit. And this kit contains a buffer, which is an aqueous buffer, and a DNA binding dye, the Qubit double-stranded DNA broad-range reagent. This is obviously in DMSO. I'm not sure exactly what it is, but it binds to DNA and becomes intensely fluorescent. And there's also 2 standards, standard number 1 and standard number 2. And so we use a Qubit 4 fluorimeter, and this is a beautiful machine. It's kind of pricey, but you can use it to measure the amount of DNA in a sample, RNA in a sample, and protein in a sample. We also need the Qubit assay tubes, and these are kind of like old-fashioned thin-walled PCR tubes. And the light path is through the tube, so always remember to write on top of the tube and not on the side. So to use this kit, what we do is we have to work out how much of the broad-range buffer we need. So for every sample, you're going to need 200 microliters of the broad-range buffer. And you add 1 microliter of the double-stranded broad-range reagent. Okay, so if you've got 5 samples, that's going to be 1 mil of broad-range buffer, and it's going to be 5 microliters of the broad-range reagent. And you add them and you mix them well. Now, for the 2 standards, use 10 microliters of standard 1 and 10 microliters of standard 2, and put them into a Qubit assay tube. Then add 190 microliters of sample buffer from stage 1, so that contains the DNA binding dye. Now for samples, you can add between 5 and 20 microliters of target DNA to the assay tube, and then again add 190 microliters of sample buffer from stage 1. I incubate for 5 to 10 minutes, and I always label on the top of the tube, as I said. And then we assay on the broad-range double-stranded protocol on the Qubit 4. So let's go to the lab and see how we do this in real life. Okay, so the first thing I'm going to do is I'm going to dilute this MaxiPrep, because it had a big pellet, and I know from experience that we need to dilute it 1 to 10. Okay, so I'm going to do 1 in 10 HA, and so I need I'll take about 90 microliters of water so we can get this kind of accurate. And then I'm going to add 10 microliters of the DNA target. So I keep it in this tube until we've got a measurement, and then we can put it on the label when we put it into the little Eppendorf. So I'm just going to add 10 microliters, so that's a 1 in 10. Got good vortex. Doesn't matter if we shear it up. Shouldn't alter anything too much. Now what we're going to do is put the standards in here. So standard 1, always put it on the lid, not on the side of the tube, because that's where the fluorescence is assay is going to take place, and you want the light path to be clear. And standard 2, okay, they're both 10 microliters, and then we're going to use the actual AH8, and that's going to be 1 in 10. Okay, so right, let's see, let's get the number 1. So this is number 1 red dot. So I've been very happy with these Qubit assays. They've been remarkably robust, and in the old days we used to use a 280-260 ratio, but that to me is kind of substandard, and when I actually ran that DNA on a gel, I had no correlation to the 260-280s that I was getting. So I moved to the Qubit, and this is much better, much better. And then this is the actual 1 in 10 dilution. 10 microliters of that. Okay. Now we want to make up the fluorescent buffer. Okay. And so what we need is a fresh Eppendorf tube, and we'll make up enough for 4. So 4 times 200 is 800. So 800 microliters of Qubit broad range buffer. Here we go. So you got the right stuff. Good shape. And then we want just 4 microliters of our dye. Again, just got it on my gloves. Yeah, now I'm just going to change gloves. Just be careful with this stuff. It's I did spin it down, but I don't think you should get it on your skin. That's really important. I don't want to be blasé with these things. They do bind to DNA. Right, so 4. Okay, so it just goes right down to the bottom. You see it there. I see it. Got a really good vortex. Okay, that's fine. So we're ready to go there. Carefully goes in the bin. And carefully put the lid back on. Get it on your fingers. Yeah, we're good. Right, so there's your mix. Now we just need to put 190 into each, and just give it a few minutes, and then we'll go to the machine. So DNA dye probably takes a couple of minutes to go on, and so we can take a little break and get the machine working. So here we go. Try not to touch anything. Here we go. Right. Go. So there we have it. So we'll see how much we've got in this mix. It could be quite interesting. Okay, so we'll go to the machine now. This is a Qubit, so it's a Qubit 4 fluorimeter. That's a very good investment. It costs about 3,000 or 4,000, but I really believe that It can be used for protein assays, for RNA assays, where it's very, very good, and for, of course, double-stranded DNA. So we're using so if we go to the home, right to the home, you want double-stranded DNA, and then you want a double-stranded broad range. Okay, and then what we're going to do is read the standards first, and then run our sample. Here we go. Here's the number 1. Just pop it in. It says read standards. Insert standard 1. We've done that. Read standard. There we go. And oh, that's very low. It's got it on the 0 there. And 2. Read standard. See, it's much higher. It's about, it's quite a bit higher. 17,000 as opposed to 169. And then we're going to read this. Remember, it's a 1 in 10 dilution. And we want a 10 microliters in there. Yeah, read tube. Right, so we have 69.4 micrograms a mil, so we've got to move the decimal point one place, so that's 694 micrograms a mil, and there's 1 mil in there, so we're kind of laughing. That's great. Okay.